So welcome everyone. Please note that we are recording this webinar so that it can be added to both our website and YouTube channel for replay. All attendees will receive a follow-up email with the link to this recording, which we welcome you to share with fellow colleagues and industry connections as you see fit. All participants will be muted during the presentation. However, we will devote time to questions and answers at the end. We invite and encourage you to submit questions at any time throughout today's presentation via the Zoom Q&A functionality. Our presenters will address as many questions live as time permits, and any remaining questions will be answered directly via email following the webinar. So I'm Satej Bogil, and I specialize in central review imaging for clinical trials on the West Coast. With experience working alongside CROs, sponsors, and research teams, I play a key role in ensuring seamless imaging workflows that support high-quality oncology research. So to quickly go over our agenda today, we'll be starting off with an introduction where I'll introduce the speakers, and we will dive right into the case studies, which will talk about retrospective and prospective studies, as well as DICOM headers, image collection, and QC as well. And lastly, we'll summarize with a who is Merit. I'm so pleased to be joined by our presenters today, Sally Assad and Alyssa Bartholomew. Sally Assad is a project manager in the oncology division at Merit and a public health professional with a background in clinical research and business development. She has earned her master's in public health from the University of North Texas Health Science Center. She gained experience and insight working in the oncology department of one of the largest hospitals in Dallas. In her role as an oncology project manager, Sally oversees the execution of clinical studies from startup through closeout, while working closely with sponsors, ensuring efficiency and compliance. She is committed to advancing novel therapies and contributing to the development of new treatments for patients. I'd also like to introduce Alyssa Bartholomew, who works as an oncology imaging quality control specialist. She is certified in general radiology, CT, and MRI, and has over a decade of experience with advanced imaging at outpatient imaging facilities, university research centers, and hospitals as well. At Merit, Alyssa ensures imaging parameters and protocol requirements are adhered to in radiographic exams that are submitted through our Excelsior software. She is instrumental in supporting study sites and ensuring image collection and quality control process running smoothly. Alyssa is very passionate about clinical trials and the advancement of cancer research through radiologic imaging. So with that, let's get things started. I will transition to Sally.
Thank you, Satej, for the introduction. Thank you, everyone, for joining us today. I'm going to be reviewing a case study that highlights how enhanced PHI de-identification contributes to tackling a common challenge that biotechnology companies as well as research sites face in the image acquisition aspect of retrospective studies. This clinical stage biotechnology company approached Merit with a need to rapidly collect and read images for a retrospective phase 2 glioblastoma study. For this study, MRI brain images were located at sites across the United States and had been performed for up to 6 years prior to Merit's participation in the study. Some of these sites face challenges with requesting the retrospective exams from their radiology departments, and in turn face delays in receiving anonymized images. Read results were needed quickly to support sponsor timelines, so this posed an overall challenge on the sites. Diving deeper into the study's needs, we identified that the major hurdle impacting site efficiency was due to the challenges that sites were presented with in terms of PHI and the anonymization process. Anonymization of the images was one of the rate-limiting steps to site coordinators. We work closely with site coordinators, and we see firsthand that they hold many responsibilities which are not limited to clinical trial, image collection, and submissions. Another limiting factor in this case was that sites were using various anonymization tools, which in turn resulted in inconsistent DICOM tags being identified during the quality control of uploaded images. The use of various anonymization tools by sites also resulted in the removal of important DICOM tags that are required for images and specified by the study image procedure manuals. As a solution, Merit implemented an automated protected health information or PHI de-identification module integrated within the Excelsior imaging platform. This module allowed for the real-time removal of PHI in uploaded images while preserving essential imaging metadata. Our quality control specialist, Alyssa, will be reviewing examples of essential imaging data to be retained later on in this presentation. Site coordinators uploaded exams into Excelsior, the imaging platform where PHI could be removed automatically by this new module. This in turn eliminated the step of site coordinators having to manually anonymize imaging prior to uploading exams into Excelsior. As I mentioned earlier, this manual process was a rate limiting step that was identified as a challenge. This solution resulted in a significantly faster turnaround time to acquire the images needed for the independent review compared to previously delayed turnaround times that were not supporting the sponsor timelines for the study. In regards to overall results and impact by the newly implemented solution, the image acquisition turnaround times were specifically reduced from 2 weeks to 1 day, which overall enabled faster retrospective data collection. This was 100% in compliance with 21 CFR Part 11 and HIPAA standards, ensuring regulatory adherence. For reference, 21 CFR Part 11 is an FDA regulation that ensures the reliability and security of electronic signatures. Excelsior has traceable and reproducible elements, and everything is tracked in an audit trail to align with this standard. The automatic removal of PHI additionally aligned with HIPAA standards to further protect patient health information in the study. This additionally eliminated site burden, including the site coordinator burden as previously mentioned, while allowing researchers to focus on data analysis rather than the manual anonymization process of the images prior to the image upload. In result, this solution overall created a standardized de-identification process. And standardization is important as it reduces variability, which can occur during manual anonymization, and ensures data consistency across different sites in the study. In this case study, I discussed the details and challenges that were faced in a retrospective study. So let's take a closer look at this type of study to better understand why the results we saw in this case played a key role in the retrospective design. In research, studies can be either retrospective or prospective. These two approaches differ in how data is collected and analyzed. And on this slide, you can see a diagram of each of these study designs. In retrospective studies, Imaging was performed in the past, sometimes several years prior to the independent review. Data is collected from existing images or records, meaning that images that are being analyzed were captured in the past. And this can limit the ability to collect standardized images from different records across different subjects. This is also a challenge that I will discuss in the next slide. In prospective studies, central image collection and potentially review is built into the study design, and images are collected as they are performed over time. This type of study design typically follows a specific protocol that is created before the study begins. Now that we've explored the basic elements of a retrospective study, let's look at some of the common challenges faced in this type of study. While retrospective studies can provide valuable insight, they can also come with a set of hurdles relating to image collection. There can be inconsistencies in how images were acquired and stored at various clinical sites, since these are images acquired in the past. Inconsistencies can also come from the loss of images if they were not stored consistently. This can also put a burden on clinical sites, as study coordinators and research coordinators need to follow up on the requests for images. It can also take radiology departments several days or several weeks to fulfill requests for imaging that was performed in the past, which results in image retrieval and manual PHI anonymization delays. For example, some smaller hospitals and clinics don't have specified departments that do this, so it may be other personnel that is working to fulfill these requests on top of their everyday clinical duties. Another challenge is inconsistencies in de-identification. There may be site-dependent variations in DICOM header modifications. For example, a series description being stripped out when anonymization is manual. This could also pose a regulatory risk, leading to potential compliance gaps in handling PHI. Manual PHI anonymization does not guarantee regulatory adherence in comparison to an automatic standard process that would ensure compliance. Lastly, there may be increased time to resolve potential queries, as it can be difficult to resolve queries on scans that were performed in the past. For example, if an image is queried in a retrospective study, a new set of images may need to be requested. In prospective studies, there are many challenges that may be faced, including things like patient recruitment and regulatory adherence. But here are some common challenges relating to the imaging component of a prospective study. One challenge includes establishing relationships with sites to ensure success at each site for the imaging component of a study. This also encompasses effective and clear communication and site support to build these relationships. As a project manager, I have learned the importance of keeping this open line of communication with sites to consistently and efficiently address any issues or gaps throughout the study. Another challenge that could be faced is information residing in multiple locations. For example, this information may include queries, images, site materials, and read data that may be stored in different locations for one study. With Excelsior, everything is streamlined and information is held in one place, and this type of challenge is minimized. Site personnel transitions are also a challenge in prospective studies. Overcoming the challenges that may come with site personnel transitions during a study would include ensuring the efficient transfer of knowledge, open queries, and imaging protocols to new study personnel, for example. Having information stored in an accessible location and being able to provide support quickly to new personnel with any transition is essential to overcome this as well. Standardizing imaging acquisition occurring at different sites may also pose a challenge in a study that is collecting images over a period of time. Again, ensuring the consistency with image acquisition and uploads across different sites is essential to overcoming this challenge. Now that we've reviewed this case study and explored the challenges of image acquisition in retrospective studies, I'm excited to introduce Alyssa Bartholomew, an oncology imaging quality control specialist here at Merit. She will be diving into the critical aspects and details of imaging acquisition and quality control, as well as image anonymization.
Thank you, Satej, for the introduction, and to Sally for the information about clinical trials. First of all, I want to go over a very high-level history of radiology. So X-ray was invented in 1895, CT was in the early 70s, MRI was in the late 70s. So as you can see, X-ray was invented quite some time before advanced imaging like CT and MRI. Then came the next phases of radiology when everything moved to digital. That includes the next two items. DICOM was invented in the early 80s. PACS was also invented in the early 80s, but not fully implemented until the early 90s. Everyone knows what an X-ray, CT, or MRI are, but DICOM and PACS are behind the scenes in every image that is being taken. To further explain, we can look at these two definitions. DICOM, Digital Imaging and Communications in Medicine, the international standard to transmit, store, retrieve, print, process, and display medical information. PACS, Picture Archiving and Communication System. This is either a program or an application where all radiology images are stored. For a long time, all images were printed out on film. For an X-ray, that's not that big of a deal because it's usually only a couple of images. But take CT for an example. Sometimes there can be upwards of 200 to 300 images per scan. All of those images used to be printed out and then taken to the radiologist to be read. With printing images comes a lot of human error. Images can get lost, they can get labeled wrong, among other things. There have been many advances and upgrades to PACS over the years that have made viewing radiology images much easier and way more convenient for everyone, including the radiologist. PACS has made image storage easier, but it has also made image transfer from facility to facility faster. Now that we know what a DICOM file is, we can explore what is within those files. DICOM tags, or headers, contain important metadata of images such as acquisition parameters, scanner device information, when the exam was performed, and patient information. Within the DICOM tag lies personal health information, or PHI. This includes data fields such as patient name, date of birth. Some DICOM tags must be anonymized to protect PHI in research, while some DICOM tags must also be retained to help with image processing. DICOM tags can only be pertinent to one imaging modality, while others are for all radiology imaging. Now that we have covered what lies beneath the image, we can explore where the images are stored at Merit. Our innovative proprietary system, Excelsior, represents a cutting-edge advancement in clinical trial management. As a single system solution, Excelsior consolidates all study data, including images, tasks, queries, site materials, analysis, and read data into one validated and secured platform. Excelsior is universal because it is a cloud-based system. This helps bypass site IT challenges when you have to download an application. Excelsior effectively manages access rights for sites, sponsors, and readers, all while having a zero footprint implementation. Our platform is transparent and very intuitive to help ease staff burden. Excelsior also supports complex studies with multiple reading criteria for different parts, and it is also FDA 510 cleared. It is important to have a software system that can recognize what tags should be anonymized and what tags need to remain. Deleting pertinent DICOM tags can lead to misinformation. For example, the series date can be anonymized by some facilities, but to Merit, this is a DICOM tag that needs to be retained because it helps us verify imaging date with clinic visit dates. Our de-identification process in Excelsior will remove PHI containing DICOM tags such as date of birth, name, and patient ID. Merit's check-in of images also ensure that no PHI gets through to the radiologist reading the exam. Although sometimes PHI will be burnt into the image, these images will be deleted by our imaging QC specialists. This is often a screenshot or a post-processing image. As you can see on the image display, the patient birth and patient ID are burnt into the image. Although the DICOM tags can be identified, it still shows on the actual image. Series description is a DICOM tag that is used for all of radiology, but I wanted to take a further look at this specific tag in relation to MRI. MRIs are comprised of many sequences or image sets. In MRI, images are scanned in 3 different planes and multiple different scanning techniques. Some sequences are more for anatomy, while some sequences are for pathologic findings. MRI is very detailed for this reason. It's also why MRI is so long. Series description DICOM tags help differentiate the images. In oncology, a T1 weighted sequence is usually scanned both pre-contrast and post-contrast for a direct comparison. So contrast will highlight or enhance certain tissues and or blood vessels to give more accuracy to aid in making a diagnosis. DICOM tags also help us identify the laterality of the image, meaning left or right. These are only 2 common examples. The series description tells us a lot of information. They help us identify the plane the image was scanned in, the type of image. They even tell us about the timing of an image in a post-contrast sequence. For example, if it's arterial versus a venous. DICOM headers help bridge the communication between the technologist scanning the patient and when Merit receives the images, especially if it's a retrospective study. For an imaging CRO, this helps us identify issues and ensures we receive a complete data set for a time point. As I said before, DICOM tags are modality specific. There are some that only pertain to nuclear medicine and PET scans. When scanning a PET, a technologists will inject a radio tracer. This is a radioactive agent. Then the technologist will scan the patient to reveal hotspots on the images or increased radioactivity. This radio tracer is very important, and that's why it has a couple of DICOM tags that are specific just for this. For SUV calculation, which is the way we calculate the uptake of the tracer, for radiopharmaceutical studies or PERSIST studies, a couple of the radiopharmaceutical related tags are injection start and stop time, dose, isotope half-life. We also need to see body, weight, height, and gender tags, and then pixel units. Moral of the story, DICOM tags tell a narrative of the how, what, and when of a radiology exam. Now we're going to move into looking at the image collection and the quality control of the images when they are received by Merit. Image storage is when images are stored centrally in one repository, organized by site number, subject number, time point, or visit name. Image quality control is when images are checked for consistency against prior time points, check for PHI, and check to make sure that they are of good quality and align with the protocol and expected imaging acquisition parameters. So automatic PHI removal. Again, this is done in Excelsior. Acquisition parameter verification for optimal response criteria assessment. That's what helps us with the reading portion. We also make sure to verify all DICOM headers, and we do a thorough review of all the images. When we do this, we are looking for lots of different things. We verify that the subject is the same one throughout all of the time points. We make sure that the dates align and ensure that all images have been submitted per the sponsor's protocol. Lastly, we can also issue queries. So queries are raised for any issue that are noted during the quality control step. This is how we communicate with sites about discrepancies. So Merit is a blinded independent central reviewer. So images are reviewed with a reading response criteria in mind. Response criteria provide a consistent way to assess tumor response across different clinical trials, enabling meaningful comparisons between the studies and the treatments. Below are a few examples of the reading criteria. RESIST, response evaluation criteria in solid tumors. RANO, response assessment in neuro-oncology. PERSIST, positron emission tomography, PET, criteria in solid tumors. And PCWG3, prostate cancer clinical trials working group 3. Each of these criteria have specific imaging guidelines. For example, RESIST uses both CT and MRI, RANO uses MRI, PERSIST will use PET, and PCWG3 also incorporates bone scans for assessment along with CT and MRI. As with anything medical, these criteria are ever evolving, so new versions of these assessments are then released. For example, RESIST was implemented in 2000, and a new version was released in 2009, which is called RESIST 1.1. This is the current standard. Response criteria can be affected by a few things in image collection of a CRO. If we don't have the full data set, or if the images are of low quality, this can make it hard to evaluate the lesions and track them throughout the studies at different time points. In conclusion, DICOM tags play an essential role in clinical research and imaging, ensuring consistency in image interpretation and data management. And now I will let Satish take over to tell you a little bit more about Merit.
Awesome. Thank you so much, Alyssa. So you may know Merit from our many years of managing clinical trials in ophthalmology. We have served biotech and pharmaceutical companies in their clinical endpoint analysis for over 10 years with a team of medical professionals and a seasoned operational support staff. We are based out of Madison, Wisconsin in the United States, but our company does have a global reach. We have team members and reading centers around the globe, making it possible to support sites and sponsors from a variety of locations. Our impactful approach is to focus on three key areas, which is our software system, Excelsior, which can handle all aspects of an imaging core lab in one single system. And this includes from acquiring images from a study site to performing an independent read, and also transferring data back to a sponsor. Excelsior can manage and track images every step of the way. Merit has a large network of over 300 radiologists and oncologists to help support your studies in a variety of therapeutic areas. And finally, at Merit, we value a partnership approach, working closely with our sponsors and sites to build partnership and long-term relationships. So Merit is more than an imaging service provider. We are well positioned to be an extension of your study team. We offer support to our partners in a variety of therapeutic areas, including oncology and respiratory. On the right-hand side, you can see that we serve as a trusted endpoint partner through four key aspects. So we leverage cutting-edge technology to streamline imaging workflows, which ensures efficiency and precision in clinical operations. Second, our team comprises of highly experienced board-certified radiologists and oncologists with extensive experience in diverse clinical trials across various medical indications. Third, we are supported by a dedicated team of project managers, imaging specialists, QA staff, and data managers. So we truly do tailor our services to meet your unique studies requirements. And collaborating closely with software developers, we can customize solutions to optimize study management. And lastly, adhering to rigorous quality assurance protocols, we ensure standardized and expertly managed clinical trials, delivering actionable insights and timely results. So our robust QA management and data privacy are upheld by four cornerstones, which include quality management being certified by ISO 13485, data security ensured by SSL, SHA-2 encryption, and ISO 27001 certification. And you also get reliability with 99.9% uptime and geodispersed data backup. And lastly, adherence to strict privacy standards, including GDP, Data Privacy Framework, and HGRAC. So Merit will be attending several key oncology conferences in the coming months, and we'd love to have the opportunity to connect with you in person. These events bring together industry leaders to discuss the latest advancements in oncology drug development, and we look forward to contributing to these important conversations. So you can find us at IO360, CMO Summit, ASCR, and ASCO. So if you're planning to attend any of these conferences, we'd be happy to meet and discuss how Merit's independent central imaging review services can support your oncology trials. So thank you so much for joining us today. That concludes our presentation, and now we will open up the webinar for questions. Please feel free to submit them through the Q&A box, and we will address as many as possible. So just got a question from the audience. So without PHI, how can you ensure images belong to the same patient that was previously received?
Great question. I can take this one. We do this by looking for several different things. We look for anatomical landmarks. So say one patient has female parts while the other one doesn't. We obviously know it's not the same patient. If they are the same gender, we can look for various things like surgical changes or missing and or abnormal organs. But when in doubt, we would always raise a query to check with the sites.
Awesome. Thank you so much. Okay, so reading another question. My company did a study 6 or 7 years ago, and we are now interested in the imaging that was done. Is it worth trying to collect the exams that were done so long ago?
Yeah, I can take this one. If these images can bring value to your current research, and it may be worth collecting those exams, you should consider things like where these images are stored, as well as accessibility. These images may be stored in different places since this is a retrospective study and you'd be going back to collect already stored images. So this is a challenge and a factor to consider.
Awesome. Okay, another question. So why do you need height and weight DICOM tags to not be anonymized?
Yeah, I could take this one. So those two specific DICOM tags are for SUV calculations for a PET or a nuclear medicine scan. So they are directly correlated with the dose of the isotope and the uptake that's within the body. Basically, it's a calculation to find the increased uptake of a tracer.
Awesome. So another question. How do sites typically manually anonymize images, and what does this process exactly look like?
So I can take this one. When exporting images through PAC systems, some sites require the images to go through an in-house de-identification process. So this can be done manually on each of the actual radiology machines, meaning the technologist can free type when registering the demographics of the patient. Manually anonymizing images can also be done by stripping metadata in image files, and sites can have different standardized procedures for this process. So this could vary depending on the site's procedures.
Okay. Another question. Is it easier to ensure that PHI is anonymized in images for a prospective study compared to a retrospective study?
Yeah, so typically, yes, because in prospective studies, anonymization is built into the workflow from the beginning with the appropriate imaging protocols. So there's more of that standardized workflow that we talked about compared to data coming from different sources like in retrospective studies. So in retrospective studies, there's also that extra step of reviewing and ensuring the removal of PHI in already acquired images.
Okay. And we will take one more final question. So are retrospective studies or prospective studies more common in oncology research?
Yeah, so retrospective studies are typically more common in oncology research, mostly due to the availability of existing data and being faster to conduct since past data is being analyzed and researchers don't have to actively enroll subjects and follow those long follow-up periods. However, prospective studies are also essential for studies that are testing interventions and tracking disease progression over time.
Let's do one last one too. So what qualifications do you have to have as a quality control specialist?
Yeah. So a lot of times you have to have a background in radiology. So for oncology specifically, we have different specialties, meaning we're board certified in different types of advanced imaging. Like I'm board certified in CT and MRI. So just radiology background.
Fantastic. I will just quickly scan for any more questions from the audience. Okay. I think those are all the questions. So, well, thank you all for attending this webinar. And as a reminder, this will be available for on-demand viewing on our website. And as mentioned, if you have any additional questions that come up later, please feel free to reach out and we're more than happy to discuss them with you. So with that, thank you so much.